Hi, today we will talk about exercise in cold climate and adaptation mechanisms that occur when exposed to cold and harsh environments. So we'll begin with hypothermia. Living and exercising in cold climates, such as northern Sweden in wintertime, is challenging and requires special considerations. In this lecture, we'll attend to most of these crucial aspects. Some of these insights and results are from the Human and Cold Network, which was an EU-funded initiative addressing some of these issues. Importantly to understand is that a man in cold is not necessarily a cold man. That is, depending on your clothing and work intensity, you may in fact overheat, also in cold climate, while at the same time get a frostbite on exposed skin. So the agenda for today is the following. We will talk about cold, hypothermia, working in different climates and different exercise modes, and then talk about the immune system and some practical implementations. So what is cold then? Well, it's a lack of heat, the sense of lost heat, lowering body temperature, and sometimes it's just defined as a temperature below -10. So now let's talk about two of the most underestimated threats in cold environments, wind and water. These elements don't just make you uncomfortable, they can drastically reduce your ability to survive and to perform physically. Starting with wind, when you're exposed to cold air, your body creates a thin layer of warmth around your skin. Wind strips that layer away, making it feel much colder than the actual temperature, a phenomenon we call wind chill. This increases heat loss, drains your energy faster, and even dehydrates you, since cold air holds less moisture. Thus, you lose more water via breathing in cold air, which is dry, compared to warm air, which is more humid. It also makes thermoregulation harder, especially during exertion, and can lead to faster fatigue and even psychological stress. So now if you add water, whether it's from rain, snow or sweat, and things get even more dangerous. Wet clothing holds or loses its insulating power, and heat escapes up to 25 times faster than when you're dry. Snow is particularly problematic due to its density. Even though you might not feel thirsty, dehydration continues through respiration and suppressed thirst cues. Of course, hypothermia risks increases. But the real danger is when you combine these two, water and wind. Think of a sleet, freezing rain, or wet snow with gusts. This combo multiplies the threats. Hypothermia sets in faster, and protective gear becomes absolutely essential. Water and windproof shells and insulating clothing are just not helpful. They are or can be life-saving. On the right now, you see a graph showing survival time in cold water. You have a 25% chance of surviving according to this gray line, and if you look at the blue dotted line, here you have pretty much 0% chance of surviving. So notice how quick survival drops as water temperature decreases. Below 5 degrees, that's plus 5 degrees, survival time is maybe just 1 hour. So for example, if the water is 10 degrees, you will most likely survive at least for about 1 hour, right? And you have about 50% chance of surviving 3 hours, but really not any chance of surviving for 4 hours. So you can talk about zones. You have a death zone, which is really where you can't survive. You have a border zone where you may or may not survive depending on different circumstances. And you have a survival zone where you most likely will survive. And this highlights how critical it is to stay dry and shielded from wind, especially during outdoor exercise or emergency situations. So my dad was fishing in Greenland in the mid-80s, and they actually had no life vests, as survival was just a few minutes if you were in the water, because it was -2 in the water due to the salt concentration. So life vests would only make them harder to get back in the boat if they fell over, and so they said. So when planning physical activity in cold environment, always factor in wind and water. They are not just background conditions, they are actually stressors that can turn a manageable situation into a life-threatening one. Look at how wind amplifies the cold and why it's such a critical factor in outdoor exercise and survival. This chart shows the relationship between air temperature and wind speed and how they combine to produce wind chill, the temperature your skin actually feels. As you can see here, even moderate wind can make you feel a lot colder and dangerously cold. For example, at 20 degrees minus, with a wind speed of 30 kilometers per hour, the wind chill drops to -33. That's not just uncomfortable, that's pretty dangerous. The color coding here highlights frostbite risk. Light blue means exposed skin can freeze in 30 minutes. Medium blue drops that time to 10 minutes. And in the purple zone, frostbite can occur in just 5 minutes. That's how quickly wind can turn a manageable cold into a medical emergency. So when you're planning physical activity in cold weather, you need to check temperature, but also wind speed, because your body doesn't care what the thermometer says. It responds to what it feels, and a wind condition that can be dangerously cold. This table illustrates how the human body responds to progressively lower core temperatures, a critical concept when discussing cold exposure and exercise. At 37, everything is just fine and normal. But even a slight drop to 36 degrees body temperature triggers increased metabolism as the body tries to generate more heat. At 35, we hit the threshold for hypothermia, and shivering reaches its peak. Below that, things start to deteriorate rather quickly. So at 34, shivering begins to fade and brain activity becomes abnormal, as you can see in EEG readings. By 33 degrees, the heart shows arrhythmias and confusion can set in. At 32, blood pressure becomes barely measurable. At 30, breathing slows dramatically, just 5 to 10 breaths per minute, and the person enters a semi-comatose state. From 28 and downward, the situation becomes life-threatening. Breathing drops below 5 breaths per minute, and the pulse may disappear. Most people are unconscious at about 27 Celsius. At 26, spontaneous ventricular fibrillation can occur, a deadly heart rhythm disturbance. At 25, the heart may stop entirely, and at 22, the brain shows no electrical activity. So to understand this progress is vital, not just for emergency purpose, but also for prevention. Once the body starts slipping below 35, time and intervention becomes critical. Explore what happens inside the body during hypothermia, not just a drop in temperature, but the cascade of physiological effects that follow. Starting with the brain, for every 1 degree drop in core temperature, brain activity decreases by roughly 10%. That's a steep decline, and by the time the body reaches 28 to 30 Celsius, coma becomes likely. This is why early intervention is critical. Now, looking at the circulatory system, cold triggers vasoconstriction, narrowing of the blood vessels, and reducing heat transport to the skin. This pushes blood towards the core, increasing central blood volume, but paradoxically, it leads to relative hypovolemia. That's because peripheral circulation is suppressed and the heart struggles to maintain output. As a result of this, cardiac output and mean arterial pressure drops. The heart's electrical activity also becomes erratic, which can be seen in disturbed ECG readings. These changes make the cardiovascular system highly unstable in cold conditions. And then there is this phenomenon known as afterdrop. This occurs during rewarming. As peripheral blood vessels reopen, cold blood from the limb returns to the core, causing a further drop in core temperature even after warming has begun. The graph on the right here illustrates this clearly. Notice how the temperature continues to fall after rewarming start. This is a dangerous phase and must be managed carefully, as people may actually die during rewarming. So hypothermia isn't just about feeling cold, it's a complex systemic threat that affects brain function, circulation, and recovery. Understanding these effects help us respond more efficiently in both critical and outdoor settings. Talk about hypothermia, right? Really talking about the body's gradual loss of control over its most basic system. So as core temperature drops below 35 Celsius, the first thing to suffer are the brain and the nervous system. People become confused, their speech blur, and coordination starts to fail. Below about 30, many lose consciousness entirely. And in very severe cases, people may even start removing their clothes, a strange phenomenon known as paradoxal undressing that is caused by a misinterpreted heat signal in the brain. The heart also slows down dramatically. That's bradycardia. And below 30 degrees Celsius, dangerous heart rhythms like ventricular fibrillation can appear. When the temperature drops under 28 Celsius, the risk of cardiac arrest increases sharply. The blood is also affected. Clotting mechanisms begin to fail, meaning that even small injuries can bleed more. Platelets become sluggish, which makes clot formation even harder. On the cellular level, everything slows down. Enzymes lose efficiency, the metabolic rate drops, and without enough oxygen, lactic acid buildup leading to acidosis. The cold itself damages cells through oxidative stress and can even trigger cell death. And finally, our immune system is affected. White blood cells stop working properly and wound healing slows down dramatically. So hypothermia isn't just about feeling cold, it's a cascade of failure affecting every vital system in the body. So when the body is exposed to cold, it shifts into energy conservation mode. Protein breakdown slows down, preserving vital resources. Blood flow is redirected from the skin to the core organ, affecting gas exchange and carbon dioxide removal. Respiration also changes. Ventilation decreases, leading to CO2 buildup in the blood. Hormone response differ and change, with stress hormones and thyroid hormones actively adjusting the metabolic rate to help maintain core temperature. So together, these adaptations help the body to survive cold stress, but also reduce overall function and efficiency. Cold exposure affects more than just comfort. It alters how gases move through the body. Vasoconstriction slows the clearance of nitrogen and carbon dioxide from tissue, which can increase the risk of decompression sickness during diving. Hyperventilation triggered by cold lowers carbon dioxide levels in the blood, but interestingly, cold doesn't change how sensitive we are to carbon dioxide itself. It just changes how it's transported. So when diving, heat plays a critical role. Heat stress during the bottom phase of a dive raises the risk of decompression sickness, which applies while applying warmth during decompression can actually reduce that risk. And finally, nitrogen narcosis doesn't just affect cognition, it also suppresses shivering and dull the sensation of cold, which can actually mask early signs of hypothermia. When we think about surviving in the cold, body size and composition plays a huge role. So let's start with fat. Fat is one of the best natural insulators. It traps heat and slows down the rate of heat loss. That's why individuals with higher body fat tend to tolerate cold exposure better. The layer of insulation keeps their core temperature stable for longer. Children, on the other hand, are much more sensitive to cold. Their small bodies lose heat easily and faster because of the high surface area relative to their body mass. Their cold tolerance is lower and their body reacts more intensely, with stronger blood vessel constriction and higher energy use. That's why kids get cold so quickly when they play outside in wintertime, even though they're running around. So if you zoom out and look across species, we find a fascinating biological pattern known as Bergmann's rule. And this rule tells us that animals in colder regions tend to be larger than relatives in warmer climates. A bigger body has a smaller surface area in relation to its volume, which means it loses heat less, quickly, at least slower. Think about a polar bear compared to a tropical bear species, or even a moose in Northern Europe compared to one in Central Europe. And of course, humans have learned to adapt behaviorally too, through clothing, shelter, and habits that help conserve heat. So evolution has given us some tools, but culture and creativity took us much further north. And here is an infographic slide of Bergmann's rule. With food and other parameters equal, a moose is on the average larger in northern than in southern Sweden, for example. From a study by Tim Noakes published in 2000, this graph highlights how body composition and activity level affects thermal regulation in cold water. On the left, we can see the thin physiologist whose core temperature drops steadily, especially when lying still. On the right hand side, the channel swimmer maintains a much more stable temperature, particularly while swimming. The key takeaway here is that fat insulation and physical activity both play protective roles. Swimming generates heat through muscle activity, while body fat slows heat loss. In contrast, passive exposure, like lying still, leads to rapid cooling, especially in the lean individual. Movement also increases heat loss by increased convection, as cold water moves energy away from the body. So for the thin person, it was better not to move, while for the more insulated person, gaining heat from the swimming, so he should move. This comparison underscores why cold water immersion can be dangerous for some and manageable for others, depending on your physiology and your movement. So summary points here. Cold is a stressor that can enhance adaptation. We'll talk more about that. It impacts performance and recovery. Energy demands increase in cold climate. You need to have protective gear and prepare for cold exercise. And cold exposure may influence immune and hormonal responses, and we'll talk more about that as well. So let's turn to physical performance in cold climate then. So here we see a competitive cross-country skier, or it's actually a ski orienteer, national championships in Sweden. And we have done some studies measuring VO2 max in cold, and in this case, in the upper right picture there, we're in a climatic chamber in -12 at Luleå Technical University, and some issues that we ran into was, for example, how to calibrate the metabolic system, as it cannot be inside the climatic chamber, and there were some discussions what temperature we should set the inhaled air to, which you need to compensate for when you do these measurements. And then at the bottom half, it's me and my dog Balder out skiing in -20. He's fine as he is, but I managed by wearing heat exchanger and correct clothing. So we'll start with aerobic and anaerobic capacities then. So this graph offers a rather fascinating parallel between animal physiology and human performance in cold environments. What you're seeing here is oxygen consumption in hummingbirds under two states, a torpid state or resting and a roused state or working, across a range of ambient temperatures. As temperature drops, metabolic demands increases dramatically in the roused state, while the torpid state conserves energy. So in biology, torpor refers to a temporary hibernation-like state where an animal can dramatically reduce its metabolic rate, body temperature, and oxygen consumption. This allows the animal to survive periods of cold or food scarcity with minimal energy use. So if you connect this to human exercise in cold climate, so just like the roused hummingbirds, humans exercising in cold environment experience elevated oxygen consumption. We can see this in increased VO2 reflecting the added metabolic cost of thermoregulation. The body isn't just fueling movement, it's fighting to maintain core temperature. The increased demand for energy affects also performance. So cold exposure reduces time to exhaustion, lowers VO2 peak, it dampens cardiovascular response like max heart rate. And the hummingbird strategy, switching to resting state, highlights the value of energy conservation. While humans can't enter torpor, as animals can do, we do adapt through clothing, pacing, and acclimatization. So whether it's hummingbird or a skier, the principle is the same. Colder temperatures amplify metabolic strain. So understanding this helps us to optimize training, recovery, and safety in cold climates. So in this section, we'll discuss human exercise in cold climate. And here are some change in performance and physiology at different tissue temperatures and skin temperatures. So if you look at the research results, they are somewhat mixed, depending on study design, with consistent findings regarding core and muscle temperatures, but when it comes to whole body exercise, such as running or skiing, many factors affect the results. So different clothing, temperatures, and even equipment calibration makes interpretation rather difficult. So there are some consistent findings, there are some different findings that differ between studies, and are in, at the best you can't interpret them. And difference in study here is clothing, wind, testing protocols, temperature used, so it's a rather difficult area to get a grip on. So with that said, here are some issues in published research summarized in this slide. So keep these issues in mind when listening to the rest of the lectures, where I sometimes have to show you something that may be questionable, so to speak, and you can look at the slide and see why it may not be correct. So the evidence for how cold affects maximal aerobic capacity is a little bit inconsistent. Some studies find that VO2 max, the maximum volume of oxygen the body can utilize, is unchanged in cold temperatures to thermoneutral or compared to thermoneutral conditions, while other reports it decreases in cold environments. When the cold is more severe, for example, as -20, VO2 max does tend to fall and participants reach exhaustion sooner. So this means that exercise performance is more limited in extreme cold, as shown by shorter time to exhaustion. During submaximal exercise, on the other hand, oxygen consumption might actually increase in cold, reflecting greater energy cost of movement and thermoregulation, or it may stay the same according to some results. So the effect of more clothing is difficult to separate from the actual physical difference or physiological difference. This graph on the right, from a publication by Ulf Berg and Björn Ekblom in 1979, demonstrates how change in body temperature, both esophageal and muscle temperature, influence maximal work time, heart rate, and oxygen uptake, all rising as temperature increases. So this confirms that lower temperature, and especially lower muscle temperature, constrain aerobic capacity and endurance through the effect, although the effect varies depending on study design and the degree of cold. When we talk about performance in the cold, one of the key questions is how our aerobic capacity, or VO2 max, is affected. Interestingly, the findings are quite mixed. Some studies show that VO2 max doesn't really change much in the cold. The body still delivers and uses oxygen efficiently, but others, like the one by Juha Oksa and his colleagues in Finland, have found that VO2 max can actually decrease when we're exposed to cold environments. In more extreme conditions, for example -20, performance definitely drops. So simply put, the body can't sustain high intensity efforts for as long when it's cold. Other factors is that more clothes are worn, increasing the work resistance and also VO2 than at a given submaximal work load. So if you look at the bottom left graph, you can see how environmental temperature influences skin temperature and performance. As skin temperature rises, both maximal work time and heart rate increases up to a point. So this tells us that warmer body generally performs better. So the takeaway here is that cold exposure doesn't have a simple effect. It depends on how cold it is, how well we're insulated, and how much the body can maintain its temperature balance. So this is from a study we did in a climatic chamber for car testing, actually, at Luleå Technical University. And I can tell you it was freezing doing these tests. On the slide, we're looking at how exercise performance and physiological responses differ when tested in warm environment at 20 degrees versus cold environment, it was at -12. In the middle, you see the primary outcome, total exercise time to exhaustion. So interestingly, the maximal performance, the time participants were able to sustain effort, was similar in both conditions. Here, they wore the same clothes in both cold and warm testing, so clothing was not an effect. This shows that cold alone does not drastically limit endurance, at least not in healthy, well-trained individuals. However, when we break down the physiological variables, there are some clear differences that you can see. Peak heart rate at the same workload was higher in cold than in warm, which is in accordance to previous studies, reflecting greater cardiovascular stress. Peak oxygen uptake was lower in the cold, meaning that aerobic power was reduced when we exercise in sub-zero temperatures. Peak blood lactate was lower in the cold, suggesting reduced anaerobic glycolysis, possibly because muscle efficiency and enzyme activity drops at lower temperatures. And peak carbon dioxide production also fell in the cold, reinforcing the observation of overall lower aerobic metabolism. So these results show that while endurance time may stay similar, our muscle heart metabolism have to work differently in cold compared to warm climates. Cold exposure stresses the cardiovascular system, and it can also blunt our aerobic and anaerobic capacity. You can see in this graph, when exercise intensity increases, submaximal oxygen consumption is consistently higher in the cold environment compared to the warm environment. What this means is that at every level of exercise, the body requires more oxygen, and thus expends more energy to perform the same work when it's cold compared to when it's warm. This increases metabolic demand in the cold and is primarily due to a combination of higher muscle activity for heat production and less efficient muscular and cardiovascular functions. The body is essentially working harder just to maintain core temperature, which comes on top of the energy demand for exercise. Statistically significant differences at each intensity increment highlights that the cold stress amplifies energy cost during exercise. And this has important implications for athletes, outdoor workers, and anyone performing physical activity in a cold climate. Greater risk of fatigue, high caloric requirements, and increased need for proper clothing and energy management. So the take-home message here is that cold exposure raises the body's energy need during exercise even before you reach maximal intensity. So here we see the responses of heart rate and oxygen uptake during exercise performed at 50% of maximal oxygen uptake at 3 different environmental temperatures. We have warm at 20, cool at 0, and cold at -15. On the left, as exercise time increases, heart rate is consistently higher in warm conditions and lower in cold. This likely reflects increased peripheral vasoconstriction and the redistribution of blood flow in the cold, leading to lower heart rate for the same submaximal intensity. However, it contradicts the previous slide with higher submaximal VO2 in the cold. On the right, oxygen uptake is shown for 3 time intervals. In every time window, oxygen consumption is highest in the cold and lowest in the warm environment, with the cool environment in between. This tells us that the metabolic cost of exercise is higher in the cold, even for the same relative intensity, probably due to extra energy needed for thermoregulation and less efficient muscle contractions. So together, these results highlight that exercise in cold environment places greater metabolic demand on the body and changes cardiovascular responses, even when doing the same work as you would in warmer conditions. So you can do the same work, it just costs you more. The body compensates for this heat loss by burning fuel and require more oxygen for the same workload. So this has practical implications for exercising or working in cold conditions, that more calories will be needed, and cold influences both perceived and true efforts during prolonged activity. So this figure highlights how cold environment affects anaerobic capacity, specifically blood lactate levels, a key marker for anaerobic glycolysis, at increase in exercise intensities. On the left, you see blood lactate concentration rising more steeply in the cold, -15, the blue line, than in the warm, which is +23, and the red line, across workout intensities, with statistically significant differences at higher intensities. Higher lactate production, or at least release to the blood, indicates that working muscle in the cold depend more on anaerobic metabolism, likely because cold or cooled muscle and reduced enzyme activity limit oxygen and efficient energy transfer. Here on the right, the main message is that when muscles are cooled, it takes a greater amount of muscle mass or higher relative effort to generate the same force or power as a warm muscle. This means that athletes or workers exercising in cold are essentially doing more work at the same task compared to in warm. Other factors such as gas exchange and enzyme activity also plays a role. So notice the practical lesson at the bottom here. Skiers are often tested in labs at room temperatures, but many competitions happen in the cold, or most competitions happens in -10 to -20. This means that anaerobic and metabolic demands can be much higher during real events than predicted from warm testing, affecting both performance and recovery. Lactate thresholds, heart rate, and training paces may therefore not match lab results. So overall, the cold increases the relative workload needed to achieve the same exercise output and raises lactate levels at the same workload, highlighting the greater metabolic strain needed for adaptation in athletes and workers exposed to low temperatures. And here is an infographics explaining the altered linear relationship between workload, VO2, and heart rate in cold climate. So these graphs show how long individuals can sustain exercise at different environmental temperatures before reaching exhaustion. At 31 degrees, representing hot conditions, the time to exhaustion is dramatically lower, indicating much faster fatigue and reduced endurance. As temperature drops towards cooler levels, performance improves, reaching its best at 11 degrees. So the moderate cold at 4 still supports good performance, almost as high as the optimal cool temperature. What this tells us is that the body performs most efficiently in cool but not freezing environments, at least for endurance events. Excessive heat impairs endurance due to higher cardiovascular and thermal strain, while moderate cold is beneficial for sustaining work over time. So indeed, if you plot marathon times versus temperature when the time was performed, so this is the average time in many different marathons, this is an important concept for planning, training, and competition, highlighting that the ideal temperature for performance is not the warmest or the coldest, but somewhere in between, a cool environment that minimizes heat stress but does not increase thermal challenges too much. So here we have an illustration of the percent difference on 3 key exercise outcomes when comparing harsh cold to room temperature. And of course, this is roughly estimated, it's not an exact number. First, we see that running time, the total duration subjects could sustain exercising, drops by more than 20% in the cold versus the warm environment. This reflects the combined burden of increased energy costs, less efficient muscle contractions, and greater thermal stress. Second is the peak oxygen uptake, which is only marginally affected, indicated the maximum aerobic capacity remains similar, but performance drops are driven by other factors. And finally, maximum blood lactate, a marker of anaerobic metabolism, or at least the release from the muscle, which is much lower in the cold. This suggests that anaerobic energy systems are less effective, likely due to impaired enzyme function and muscle metabolism in the cold, which limits high-intensity performance and accelerates fatigue. So altogether, these trends highlight that while aerobic capacity stays nearly the same, actual work capacity and anaerobic function suffered very cold temperatures, resulting in significantly earlier exhaustion and reduced performance in real-world cold exposure. Then don't forget that the increased strain from heavier clothing in cold environment has an effect on performance. Okay, let's turn to muscle function, force and power. When we look at neuromuscular function, several functional aspects of movement and performance are negatively impacted by cold tissue temperatures. As shown here, force generation measured in newtons and muscle power in watts and movement velocity, usually measured in meters per second or degrees per second, all decline as muscle and nerves cool. Coordination is also reduced, as cold impairs not just muscle but also joint mobility and nerve conduction. On the right side, you see the thermometer as before, illustrating how progressively decreasing tissue temperatures lead to specific losses. Below 28 degrees, muscle strength, power, and endurance start to drop. Joint mobility decreases under 28 degrees Celsius, compounding movement difficulties. And then nerve conduction slows at around 20, reducing reaction speed and muscle control. And fine manual dexterity suffers below maybe 15 degrees Celsius, affecting skilled movements, like fine finger control and such. And under 8 degrees, tactile sensation fades, ultimately risking numbness or loss of feeling, and you can't really feel what you're holding. So these changes mean that cold not only makes movement harder and slower, but also places individuals at risk for slipping, mistakes, or injuries due to impaired coordination and sensory feedback. This is critical both for athletes and workers in cold environments, highlighting the importance of maintaining adequate muscle temperatures to preserve safe and effective movements. Exposure substantially alters EMG, electromyography signals in working muscles. In the cold, most muscle groups show reduced EMG activity or greater gaps as this is measured during repetitive work, reflecting impaired muscle recruitment and coordination. And the timing and amplitude of muscle activation also changes as seen in these waveforms. So this was a study on working in cold environments. Clothing helps mitigate some of these negative effects, but cold still compromises neuromuscular function. So overall, muscle performance and control decreases in cold, increasing fatigue and reducing fine motor skills. Illustrates how maximal force production, measured here in newtons, increases as environmental temperature rises. In cooler temperatures, 10 degrees to 15 degrees, muscle force is significantly reduced, indicating impaired strength and power. As temperature approaches room temperature, 20 and above, maximum force improves, peaking at 27. In fact, I think that it continues to increase if muscle temperature goes up to around 39-40. And how does this affect performance then? Well, colder temperatures sharply limit the ability to generate force, which directly reduces athletic and occupational performance in tasks requiring strength, speed, or power. So maintaining warmer muscle and tissue temperatures is crucial for optimizing performance, especially in strength-based and high-intensity activities. Cold not only makes muscle weaker, but also increases the risk of injuries and decreases endurance, as we saw before. This figure shows that maximum voluntary contraction, MVC, a measure of maximum force or muscle force, declines more rapidly during submaximal work in cold compared to warm conditions. Over 2 hours of repetitive low-intensity work at 10% of MVC, max MVC dropped by 28% in the cold, but only 15% in the warm. So how does this affect performance then? Well, cold significantly accelerates loss of muscle strength during prolonged work. This means working in cold environment causes much greater muscle fatigue and functional strain even at the same workload. Individuals will tire more quickly and be less able to generate force, increasing risk of errors and injuries. Maintaining muscle temperature or limiting exposure time is therefore critical to sustain safe and effective performance in the cold. And this is of course also true for workers. And here is a graph to demonstrate some maximum muscle power decline over time at different ambient temperatures. At 10 degrees, which is cold or cool, power starts lower and falls faster compared to both 27 and 40 degrees. Power loss is most pronounced in the cold, highlighting the cooler environment not only reduces peak muscle power, but also accelerates fatigue and performance drop during sustained activity. So looking at sports then, sport performance, we can see that prolonged exposure to cold causes a faster and greater reduction in maximum power output. Power is often more important for performance than force. This means athletes and workers in cold environment will see their speed and force diminish more quickly, increasing risk of errors and slowing task completion. And it also affects recovery unless temperature is managed. This figure shows that throwing velocity is consistently lower in cold versus warm conditions across different ball weights. As the ball gets heavier, cold temperature impairs throwing speed even more, with significantly differences at all measured weights. So affecting performance then, cold muscle and tissue temperature reduces speed and force the force athletes can generate when throwing objects. And this of course impacts sport performance, occupational tasks, and activities requiring rapid, powerful arm movements, especially when handling heavier objects. Warmer conditions can help preserve muscle velocity and high-speed coordination. So of course it's important to keep the muscles warm. Specific performance then. And these slides illustrate how cold environment affects sport performance across different disciplines. So starting with football or soccer, despite common assumptions, running distance during matches remained relatively stable across temperatures. And this suggests that players maintain their physical output even in colder conditions, likely due to pacing strategies, match demands, and just keeping the muscles warm. In cross-country skiing, however, we can see a drop in power output at -14 degrees compared to 0. This indicates that extreme cold may impair muscle efficiency or coordination, even in well-trained athletes. Also remember that cross-country skiers can't really warm, and they have this really tight racing suits. Interestingly, heart rate is slightly lower in the colder condition, which could reflect reduced cardiovascular strain or altered pacing. And lastly, running economy improves as temperature drops, meaning athletes use less oxygen to maintain a given pace, but to a certain degree, of course. Cooler air reduces thermal stress, which may enhance efficiency, especially in endurance sports. As we could see in the previous slide, this indeed transferred to marathon running times. So overall, cold doesn't uniformly impair performance. The effect of cold depends on the sport intensity and how well athletes adapt to strategies and physiology. So this slide breaks down how cold environment affects sport performance across 3 layers: physiology, capacities, and actual performance, all based on data collected at 21, 10, and -10 degrees Celsius. So let's start with physiology. As temperature drops, rectal temperature first increases, then decreases when temperature drops below 10 degrees. Water loss measured at lost body weight decreases as temperature drops. You sweat less, and perceived exhaustion is apparently highly individual, as you can see on the standard deviation in the bottom figure. Moving into capacities then, we can see that VO2 max stays constant while peak heart rate declines in colder conditions, indicating reduced aerobic capacity, yet not affecting VO2 max. And on the anaerobic side, max lactate decreases with temperature, but lactate when running at the individual lactate threshold speed remains constant. Finally, if you look at the performance outcome, running speed at the 4 millimole lactate threshold and running economy did not change. So you see that the VO2 max per kilogram body weight did not change. Time to exhaustion did not change significantly over time, but the trend of an optimal time to exhaustion around 0 to +10 has been seen also in other studies. Important to note in all these results is the large variation or standard deviation, indicating very individual response to cold. So in summary, cold environments affect some variables in some individuals and may for some athletes in some conditions limit both capacity and performance. These effects highlight the importance of acclimatization, warm-up strategies, clothing, and pacing in cold conditions. Combining the effects of cold with the need for protective clothing, performance drop can be estimated, as shown in this table created by Juha Oksa at the Finnish Institute of Occupational Health. So then, let's move on to the immune system. So let's look at the effects of the cold on the immune system then. And this slide summarizes the key effects of cold exposure on immune function. So first, cold activates stress hormones and suppresses immune activity. You can have barrier damage when dry and cold air weaken mucosal defenses in nose and throat, increasing infection risk, or at least increases the possibility of pathogens entering the body. The cytokine response and immune cell function are impaired, so immune signaling and recruitment can slow down. You have a change in blood flow or vasoconstriction, and microbiota composition further undermines health. So you have seen that microbiota in the cell, in the gut, actually changes. You have a decline in immunity is reflected in higher rates of respiratory conditions such as COPD and general susceptibility to infections, especially during colder months. But we'll look at some new data on that in a second. Overall, cold exposure challenged the immune system through both direct physiological impact and is suggested to increase the likelihood of infections. This suggestion is, however, a challenge, and we'll come back to that in a second. So cold exposure alters both innate and adaptive immune markers in humans, but findings are context dependent and sometimes conflicting. Acute severe chilling or hypothermia has been associated with suppressed cellular and humoral response in animal models, and with increased respiratory infections risk in humans. Repeated or controlled cold exposure can mobilize leukocytes and raise anti-inflammatory mediators. So this table summarizes common patterns across exposure types. As you can see here, the effect of cold environment on the immune system is rather complex, and we're not going to go through all of this, but if you're interested, you can look at these publications. So what I've done instead is to summarize the key findings in a visual image here. Overall, the cold stress modulates adaptive immune responses, sometimes stimulating and sometimes suppressing them, depending on the intensity, duration, and frequency of exposure. So the key findings here is that mild or short-term cold exposure, for example, swimming or slight temperature drops, can stimulate immune activity, increasing proliferation and cytokine production like IL-2, IL-4, and other cytokines. Severe or prolonged cold stress, for example, -20 exposure or chronic cold, tends to suppress immune responses, reducing proliferation, antibody production, and delayed type hypersensitivity responses. And different immune cells, B cells, T cells, and other lymphocytes respond differently, indicating a complex, context-dependent immune modulation. Cold stress exerts bidirectional effects on the innate immune response, with outcomes depending also on exposure intensity, duration, and also different by different organisms. On the previous slide, which is inserted a little bit smaller here, we saw theoretical and experimental evidence that the effects of cold on the immune system depends both on severity and duration of exposure, as well as immune cell type. Mild or brief cold was shown to potentially boost immune cell proliferation and cytokine production, while severe or prolonged cold suppresses immune function. However, when we look at this slide, with data from a whole body cryotherapy study, we can see inconsistent and sometimes contradictory results. Some markers, such as lymphocyte proliferation, and cytokine production do not show the expected increase or decrease after cold stress, even after 9 weeks of repeated cryotherapy. And similarly, the innate immune markers, like monocytes and granulocytes counts, appear largely unchanged throughout the intervention. This suggests that in real-world settings, or at least with the specific cold exposure protocol used in this study, the bidirectional and context-dependent immune effects highlighted in the literature may not fully translate into measurable changes in blood biomarkers. In other words, while the overall model predicts variable immune activation or suppression depending on condition, the observed results in specific studies can be inconsistent, reflecting the complexity of the immune regulation and the influence of many confounding factors, such as cold acclimatization, individual biology, timing of measurement, protocols used, and what biomarkers cytokines are measured. This mismatch points to the ongoing challenge in validating cold-induced immune modulation and the need for more robust research designs and biomarker panels to truly capture these adaptive or suppressive responses. So this slide highlights one of the most debated topics in environmental physiology, how cold exposure affects the immune system. Put your hat on, you'll catch a cold, my mom always told me, and probably other mothers as well, and fathers. So let's start with the first point, immune function. So contrary to popular beliefs, tightly controlled laboratory studies show that acute and repeated bouts of cold exposure can actually stimulate the immune system. This means that short, deliberate exposure, like cold plunges or winter swims, might activate certain immune pathways and enhance resilience. It's a hormetic effect, a small stress that strengthens the system. But the second point, infection rate, reminds us that context matters. The widespread belief that cold exposure, especially when combined with exercise, suppresses immune function and increases the risk of upper respiratory tract infections. And that belief isn't unfounded. As we've seen in earlier slides, cold stress elevates cortisol and noradrenaline, which can blunt leukocyte activation and cytokine signaling. Add intense physical exertion and the immune system may temporarily be compromised. So how do we reconcile these two perspectives? Well, it's all about the dose and duration parameters. Brief, controlled cold exposure may be beneficial, but prolonged or extreme cold, especially during intense exercise, can tip the balance towards immune suppression. This is particularly relevant for athletes and military personnel or other outdoor workers in northern climates, like us up in northern Sweden. In summary, cold is not inherently harmful or helpful, it's a tool. Used wisely, it can enhance immune function. Used carelessly, it can increase vulnerability. Understanding these dualities help us design smart training, recovery, and health strategies in cold environments. To more population-based studies. This slide presents evidence on how cold exposure contributes to increased mortality in cold seasons. On the left panel, you can see mortality by month, unadjusted, and then the bottom panel is adjusted for month, low temperature, and influenza, which is rising in the winter months. On the right side now, you can see a very complex panel, and we see long-term scatter plots of excess winter mortality, EWM, over decades, showing some variation and a rising trend in recent years to straight lines. So it can be concluded that many confounding factors influence mortality rates. And this is something called winter death. So this quote published in, or the article I should say, published in National Geographic a few years ago, highlights the excess winter mortality, a pattern seen repeatedly in data, reflecting a complex balance of long-term trends, including not just environmental cold, but also contributors like influenza outbreak, vaccination rates, population aging, housing quality, and healthcare cost. And expanding this slide on the mortality trends associated with cold exposure, emphasizing that winter deaths are the result of multiple interacting factors rather than a simple temperature alone. The black line is the simple mortality rate. The red and green lines, mortality rates when many other factors are compensated for. So it's 8 or 6 factors here. The cold temperature have no effect, while increasingly warmer temperature above 20 degrees Celsius remain a factor increasing mortality even when compensating for other factors, confounders. And here we can see a simplified graph of how the effect of cold per se, or temperature, disappears when other factors are added. Heat, on the other hand, kills more people than other confounding factors accounted for, as can be seen by the sharp increase from 20 to 30 degrees. Here's an infographics summarizing the winter mortality concept. So, moving into adaptation to cold. Can we get used to living in the cold? When I first searched PubMed for publications on this, I only had 23 hits, which means that this area of research is under researched, not too many studies on this. There are 3 main types of cold adaptations. You have a metabolic, the raise in metabolic rate and heat production. You have insulative, reducing heat loss via vasoconstriction or fat deposition. You have a hypothermic, lowering the threshold for shivering and cold discomfort, so you can sustain the feeling cold. And key mechanisms include activation of brown adipose tissue of bats to produce heat without shivering. You have increased sympathetic nervous system activity, leading to vasoconstriction and higher metabolism, and beneficial effects from repeated cold exposure that improves perception and tolerance. If you look at dogs, for example, they shiver when it's really cold. It's not because they freeze, it's because they generate heat to avoid freezing. So examples here are traditional pearl divers in Japan and Korea who spend long periods in cold water with minimal protective clothing. They get used to this. Fishermen and cold water workers who develop heightened tolerance through their occupations. And indigenous cold climate populations who rely both on some physiological changes and extensive behavioral strategies such as clothing, shelter for survival. So overall, while some biological adjustments do occur, human cold adaptation is dominated by habits, technology, and culture. This is in contrast to animals in cold climates, which often exhibits profound physical adaptation to cold. So, as an example or a focus here is this protein UCP1, which stands for uncoupling protein 1. It's located inside the mitochondria of brown fat cells. And this UCP1 has a unique ability to uncouple oxidative phosphorylation, the process normally used to make ATP, and instead it directs this energy to generate heat, a process called non-shivering thermogenesis. This mechanism enables animals, and to a lesser degree humans, to stay warm in cold environments by burning stored energy for immediate heat rather than movement and storage. UCP1 is therefore essential for maintaining core body temperature during cold stress or fasting, and is a key target for understanding evolutionary and genetic adaptation to cold climates. So in summary, evolutionary changes in UCP1 as well as its activation are vital for efficient heat production and survival in the cold, making this protein a central player in thermal biology and metabolic health. This diagram shows the mechanism by which UCP1, the uncoupling protein in brown adipose tissue's mitochondria, creates heat through non-shivering thermogenesis. Importantly, genetic differences in the UCP1 gene, represented here by the allele T to the left and allele C to the right, can impact this process. Individuals with the T allele may have greater amounts of UCP1 or more efficient uncoupling, resulting in higher heat production and better cold tolerance. Those with the C version may produce less heat, making adaptation to cold more challenging. This distinction demonstrates that the evolutionary importance of UCP1 for surviving cold climate and help explain differences in thermogenic capacity observed in human population and animal models. The complexity of thermogenesis and also the adaptation to cold in human and animals. Starting on the left, cold exposure stimulates the hypothalamus and activates the sympathetic nervous system. This leads to the release of norepinephrine and endocannabinoids, which triggers signaling pathways in brown and beige adipose tissue via beta-3 adrenergic receptors. A cascade of downstream effects follow, and other signaling molecules cause lipolysis, releasing of fatty acids, and other effects. Within the nucleus, various genes, most importantly UCP1, are then upregulated in response to key metabolic and hormonal signals. Finally, in the mitochondria, UCP1 uncouples oxidative phosphorylation, allowing protons to flow across the mitochondrial membrane without generating ATP, instead producing heat. This figure highlights just how multi-layered cold adaptation is. From brain signaling to gene regulation, fat breakdown and specialized organelles all work together. The ultimate evolutionary outcome is non-shivering thermogenesis, efficiently converting energy to heat rather than storage in response to cold environments. Cascade of downstream effects that begin with UCP1 activation, the key protein for non-shivering thermogenesis in brown fat, I should say. Once UCP1 is active, it doesn't just produce heat but affects a wide array of signaling pathways. These include thermogenesis itself, mitochondrial function, metabolism, fatty acid cycling, and respiratory electron transport. So, significantly, UCP1 links to important processes far beyond temperature regulation. The cardiovascular system, angiogenesis, the formation of new blood vessels, overall metabolism, maintenance of homeostasis, and many fundamental cellular functions. This illustrates that cold adaptation, mediated by UCP1 and its related metabolic shift, influences not only how efficiently we can produce heat, but also impact health, disease risks, energy balance, and stress physiology throughout the body. This integrated figure highlights how variation in the UCP1 gene, specifically the TT genotype, assists human adaptation to cold climate across the globe. At the left here, you see a world map showing the geographic distribution of the T allele, and it's more common in population from colder regions, consistent with the evolutionary selection for cold tolerance. Supporting data below the map show that people with the TT genotype have higher frequency in colder populations, or people living in colder areas, enhanced metabolic activity and greater ability to generate heat via brown adipose tissue, and a more robust thyroid response shown by the FT3 and thyroid hormone values. They have a larger body surface area, which may further aid heat generation and retention. So the overall finding is that specific genetic variants of the UCP1 promote survival in cold environment through biological and hormonal mechanisms. This pattern of UCP across the globe strongly suggests natural selection, favoring traits that support efficient thermogenesis, better metabolism, and greater adaptation to environmental cold. These shifts are visible on the population level, linking genetic, physiology, and geography in the story of human cold adaptation. As stated before, relatively few well-conducted studies in human cold adaptation are published, which is obvious when looking for meta-analysis. There are very few meta-analyses on this subject. Here we see how even relatively mild cold treatments, exposure to 18 degrees on the naked skin, can affect energy metabolism in adults. Here it's important to know that 27 degrees is the thermoneutral zone for a naked human, so 18 degrees demands a slight increase in metabolism. On the left here we see several studies consistently demonstrated an increase in energy expenditure when subjects moved from room temperature to mild cold. And on the right side, multiple studies report significant increase in brown adipose tissue volume under the same conditions. So taken together, these effects illustrate that mild cold exposure may be enough to trigger activation and growth of brown adipose tissue and ramp up energy usage and heat production. This response is beneficial for thermogenesis and may play a role in metabolic health and weight management. Overall, cold adaptation is a dynamic process that can be activated by small changes in ambient temperature with measurable outcomes for both energy expenditure and bat biology. This overview shows potential ways in which repeated, carefully managed cold exposure may promote longevity and counter some of the major changes associated with aging. Now, this is highly hypothetical. It's up for research, I would say. So inflammation. Cold exposure is known to reduce key markers of inflammation, boost anti-inflammatory cytokines like IL-10, and generally stimulate aspects of immune response. On cardiovascular health, then you have regular cold stimulus can increase cardiac output, boost vagal tone, an indicator of health status, and improve peripheral circulation. You also have metabolic regulation, where cold activates brown adipose tissue, BAT, increased daily energy expenditure, improves the efficiency of metabolic processes, and support better insulin and glucose handling. And on the topic of oxidative stress, there is evidence that cold exposure boosts the body's antioxidant defense, lowers the production of reactive oxygen species, ROS, and increase protective enzymes like SOD, and help regulate uric acid levels. While these results are promising and supported by emerging research, they are most robust in experimental settings. Translating them into practical anti-aging interventions for humans will require further studies and careful application. And as I said before, there are few studies on this topic. So take-home messages then from this lecture is we have talked about what cold is, the limits for cold, what is hypothermia, when the core body temperature actually falls. We have talked about work capacity in cold, muscle function and coordination, how that is affected by cold environments and when the tissue is colder than normal. We have talked a bit about the immune system and how cold can both suppress and enhance immune function. And then finally about long-term adaptation. So the take-home message for then about the winter death rates, which has been fairly consistent, but this last studies show that there are many factors accounting for this increased winter death, not just temperature. So if you look at the NIH recommendations for personal protective equipment and working in the cold, you have a infographic slide here. And I also have some personal tips, since I have been born and raised around the Arctic Circle. I was born above the Arctic Circle and grew up slightly below, but I have been out running in -27. It's been slow and not so far, but the training was done also that day. So here are some tips if you want to go out and try this. Thank you for today.